Okay, so the ratio of 9, okay, is to 3:4, wherein 9 will be all a gouty, which is gray. 3 will be all colored, and 4 will be albino, white. Okay. Let's come to the next one. Dominant epistasis. Dominant epistasis, okay. Dominant epistasis here, the example would be in summer squash. Example would be in case of summer squash. Cucurbita papo. Cucurbita papo, okay. Now in this case, if you see white fruit, okay, you get a white fruit if you have these possible combinations. Both these combinations give you white. You end up getting yellow if it is this combination. Okay. Whereas you get a green fruit if it is recessive. So you have a precursor molecule. Now this precursor molecule, which is supposed to be green, okay. Now this can give you, now in case if there is W, okay, if there is W, you end up getting this green color, okay, will give you a white color if you have a W gene. If you have a Y gene, okay, then it ends up giving you yellow. But if you see W, okay, when this is present, you still end up getting white. So this capital W which you have, okay, that masks the yellow. So W is dominant over yellow. Yellow is also a dominant gene, but who is masking the effect of another dominant gene? W. So W masks the effect of Y gene product. All right. So because of which when only W is present, this gene product, so you will never get a yellow. So both these combinations, this or this, can give you a white color. So when, okay, you have, even if a capital Y is present, can you see here? Even if a capital Y is present, you will never get a yellow color, but you will get white. But when can you get a yellow color? Only when this capital W, or only when this, when this W is recessive. Only then you end up getting a yellow color. Okay. So W masks the effect of capital Y. Perform a cross of white with a green. This is green. These are whites. Okay. Of course, the progeny that you get here, F1, you end up getting hybrid. This is going to be a white. Report to be the F2. So dominant epistasis is when a dominant gene, which is capital W, is stopping the expression of yellow. So when both are present, white is getting expressed. Yellow won't be expressed. Okay. Only when W is recessive, that's when yellow can be expressed. So this ratio, what you end up getting here is 12:3:1. So 12 white, 3 yellow, 1 green. Okay, so 12 will be white, 3 will be yellow, and 1 is going to be green. Okay, so coming to polygenic inheritance, which is also called as quantitative inheritance. Now, what happens in quantitative inheritance is the dominant genes basically have cumulative effect, and the dominant allele expresses a part of the functional polypeptide, and full trait will be shown only when the dominant alleles are present. Now, example I would say is, for example, okay, let's say I have capital A gene, okay, and capital A gene. And let's say this is responsible for a height of an individual. So let's say when it comes to like, and basically when I speak of polygenes, I cannot speak of just one gene. I'm supposed to be speaking of more than one genes. All right. So let's say that, let's say, skin color of a person, or height is one example. Then skin color, you don't have just black and white, correct? You have different range of the skin color. So let's say I have A, B, B, and C, C, and this is responsible for, let's say, like, very light colors. So person who's like extremely fair has got this gene combination, and let's say to get me a light color, I need about 60 units of these genes. Okay, so if 60 units, then don't you think I have 1, 2, 3, 4, 5, 6, then each gene is going to contribute, or each allele will contribute to about 10 units. And that's how I end up getting a lighter color, which has 60 units. So any person, okay, who's dark, who, let's say, for example, is very dark. Let's say the combination, okay, so I can give it either like, let's say small a, small b, or small c. Okay, or I can, give it genotype. This is just an example I'm giving here. So let's say for this very dark combination, let's say I need very less units. So in case if it is recessive, let's say this is giving me a very dark combination. All right. So in case if I have this combination, heterozygous for all, so 10 units of by this, 10 units by B, capital B, and 10 units by C. So don't you think I will have a different, a mid-range, let's say wheatish color. Let's say for a wheatish color, I will need only about 30 units. And let's say for a dark color, I just need 0 units because none of the genes are contributing to it. So here with 30 units, don't you think I will get a mid-range color? Okay. So it all depends upon like, how many units are contributed by each genes. That's called as, because of which we call, height and skin color as quantitative inheritance. All right. So what is quantitative inheritance? Wherein each dominant allele expresses a part of the functional polypeptide, and full trait will be shown only when dominant alleles are present. So genes which are involved in such quantitative inheritance is called as polygenes. Okay, so these are all the polygenes. So many genes are responsible for a particular trait. So this was just an example that I gave you. Okay. So when it comes to height of a person, or skin color, okay. So let's work out, with regards to the skin color. For skin color, let me give you this example here. Let's say a person who is white, okay, and let's say who's black, negro. Okay, for white, I mean, I'm just changing the units now. For black, I'm giving them, let's say a gene is contributing to a dominant gene is contributing to black. So here, let me put it as recessive conditions for all, and here I'm putting it as dominant genes for a black color. Okay, because it is responsible for pigmentation. So this, let's say, is a very dark combination here. F1, what I end up getting is heterozygous condition. Now what do you end up getting here for heterozygous condition? This is intermediate color, intermediate. And these intermediates are called as mulattoes. That's a mulatto. Intermediate color. So when you do such a tri-hybrid cross, okay, when you do such a tri-hybrid cross, your phenotypes will be 1:6, okay, wherein 1 will be very dark. This is going to be dark. Different shades you'll get. 15 will be fairly dark. Because different units will be contributed, correct? 20 will be intermediate. 15 will be fairly light. Fairly light. 6 will be again light. And 1 will be very light. So this is going to be very dark. Okay, so when you speak of skin color, you usually end up getting a graph wherein the intermediates, okay, intermediates usually will be at the top of the peak. So this will be contributed by your intermediates. Very dark, so very dark and very light will be towards the extremes. Okay, intermediates will be high in number. Okay, so you end up getting a bell-shaped curve. So these are 3 pairs of polygenes. So more than one gene is responsible for the same trait. Okay, and the expression of the trait will depend upon how many dominant genes are present. Okay, so one more example would be kernel color in wheat. That is also polygenes. Okay. So let's come to sex determination. Now, first is basically, okay, under sex determination, this is chromosomal basis of sex determination. Okay. Now, first, we'll start with A. We'll start with male heterogamety. Male heterogamety. Okay. Now, when it comes to male heterogamety, we are speaking about the first one, which is the XO type. XO type, which is also called as the XO type. But this is basically seen in insects. Okay, this is seen in insects like grasshoppers and all. Now, what was seen is basically okay. So when it comes to these insects, they have autosomes, okay, and they have sex chromosomes XO. So if it is only one and the other one is missing, okay, this happens to be a male. And if you have autosomes and you have 2 X chromosomes, then this happens to be a female. Now, these autosomes of a pair in a male, they separate out into autosomes plus either X. This is one gamete that you get. Okay. This is one gamete that you get. And the other gamete that you get in a male will be autosome plus nothing. Nothing is nothing but the O type, we say. O0 or O type, which is no X chromosome. Okay. So these are the gametes. Whereas in a female, you have only one type, which is autosome, one of the autosomes plus X. Okay, so this is going to be these are going to be your sperm, which are going to be 2 types. Whereas the egg will be just one type. Because there are 2 types of sperms, we call it as heterogamety. Okay, so this is male heterogamety, which is seen in insects. So 50% of the sperm, if you see, 50% of the sperm will be different and 50% of the sperm will be different, wherein one will have only autosomes, okay, and one will have the X chromosome. But eggs, females will have all the same type, 100%. This is female homogamety. coming to the next type, okay, wherein XY type, which is XXXY type, seen in humans and Drosophila. Males have XY type, females have XX. So females gametes will be 22 plus X, males will be 22 plus X or 22 plus Y. So heterogamety is seen in males. Okay. So when it comes to in males, as I told you, that is gametes could be X and X, females could be X and Y. Okay. So X with this X or X with this Y, you could get a male and a female. So this X with this X and this X with this Y, you could end up getting a male. So there are 50-50% chances of getting a male and a female to a couple. Now let's come to female heterogamety. This is also the ZW type. We call it as ZW, ZZ type. Okay. So the moment you have, ZW, this is a female, and ZZ would be a male. Now this is seen in case of birds. Okay. So Z and W, this is a Z type. So the moment you have Z and Z coming together, that's going to be a male, and Z with a W will be a female. So females show heterogamety. So females show heterogamety. In case of birds. So under female heterogamety, it was ZW, and the second one is ZO type, which is ZZ ZO type. This is seen in butterfly and moth. Butterfly, moth. Okay. So basically the females, they show two types. So ZO and ZZ. ZO will be a female. This is going to be a male. Okay. So females show autosomes plus Z, or they can show autosomes plus O. Okay. So heterogamety is seen here. In case of females, this will be A plus Z types only. Males. So females basically are ZO types.